And also, this is the exam that we took together, okay, which will be our example for today's session regarding the experiment, okay. The experiment I gave you was this one. It was out of 6, okay. This is number 1. The second skill is the skill of making a table. The table is vary from 3 to 6. The minimum number or the minimum grade that came in the past 3 years was 3, and the maximum was 6 marks given to the table. So it's a very important skill to know it and to know how it will be graded. Number 3 is the drawing. Drawing, I mean the one that maybe a lot of you, I'll not say all of you, but many of you, okay, confused a little bit how it will be graded. Did I do it correctly? Okay, so we will go through it. And also it's not less than 3 and not more than 5, okay, in all of the past years. The last thing is the graph. And a graph is vary, maybe a line graph, maybe a bar graph. Those are the most things that occurred or that frequently came in the last 3 years. So we're gonna start by talking about the designing an experiment, okay? First of all, any experiment should be contained variables. Any experiments should be contained variables. So experiment, if we are saying, for example, experiment, any experiment, okay, should be containing the variable. The variables are categorized into 3 categories. Category number 1 is the independent variable. This is the one that we are changing by ourselves. This is the one that we are changing by ourselves, okay? So this is the first thing, and you have to write it in the first, okay? Second, we have the dependent variable. The dependent variable is the one that is changing according to the independent variable. So this is the changing variable. The changing variable. So the variable number 1 is independent variable, the one that we change by our own, and mostly we are studying this, okay, the effect of this on, or the effect of independent variable on the dependent variable. Variable number 2 is the dependent variable, or the changing, the measuring one, the one that we measure, okay. And lastly, we have the controlled variable. Controlled variable are all of the other variable, okay, that should be kept constant during the experiment, okay. All of the other variables that should be kept constant throughout the experiment, okay. So we have 3 variables. Number 1, independent. Number 2, dependent. Number 3, the controlled. Regarding the marking or the grading for the 3 variables, always the independent variable is for 1 mark, and the dependent variable is 1 mark, and the control variable, it's vary from 2 to 3 marks. 2 to 3 marks. Minimum marks given is 2, maximum is 3. So the first point is half or even more than half of the experiment, the variables. If you determine them well, okay, you will do the experiment correctly, okay. So for example, if we look at this question that we did in the exam, student was asked to test the hypothesis, which says or stated that catalase activity is the same, okay, in all species of plants, okay. So he asked you to plan an investigation to test this hypothesis, okay. If we back to the mark scheme as or upon with you graded, we have here independent variables, okay. You should mention, here he mentioned at least 3 different species of plants. 3 is the minimum, but the ideal according to the work of the experiment is 5. 5. Always mention 5, okay. 5 is better. 5 is the ideal. 3 is the minimum, okay, or at least, but 5 is the ideal. This is the independent. Dependent variable, okay, in this case, okay, measure height of bubbles or the volume of oxygen produced, okay, and then you can count the bubbles. So this is the dependent variables. the dependent variables, and also graded for 1 valid marking point. If we together look here for the controlled variables, okay, you can see that the controlled variables maximum to from, which mean the maximum grade here in this specimen or in this paper, okay, is, or this variant is 2, okay, grades or 2 marks. You can mention any of those, okay. Those are things that should be kept constant during the experiment in order not to affect the results, okay. Here, for example, same size, mass plant, same volume, and so on, okay. So now we discuss together the variables, the independent, the dependent, and the controlled. Those are more than half of the marks of the designing an experiment, which is must come in all of exams with minimum 5 and maximum 6 marks. So after this, we can talk about the methodology, okay, or how you can handle the experiment, how you can run the experiment, okay. This is also graded from 2 to 3, okay, and in some cases 4 marks, okay, rarely 4 marks, but mostly 2 to 3 marks are being given to the methodology or the way by which you are doing the experiment, okay. Yeah, any method, okay, you have to give details of the method that you will go through in order to find out the results or to measure the dependent variable, okay. So just a while, okay, now 1 second. Okay, 1 second please. Okay, let's back. There is one that annotates now the on the screen. I wish not to do this, or instead I will block all of the annotation. Okay, I don't want to do this, by the way. So back. Okay. Okay. Now let's back to our experiment. Okay. Just a while. Okay, now I did already to prevent the annotation from the from your side, because actually there are some students that misuse it. Okay, back. Now we started together the number 1, the variables. We have independent, dependent, and controlled, and together they are making more than half of the mark. Okay, the method is being graded from 2 to 4 marks. Okay, so try to write a consistent, relevant, logic, consequent steps in order to gain marks. Okay, and ending the methodology always by calculating, by measuring, by taking the results or the reading of the dependent variable, the changing one. Okay, so if we mention, for example, the marks for independent is 1 and dependent is 1, and the method's minimum is 2, so 2 plus 2 are 4, remaining are 2. So the methodology will take 2 at least, so it will be 6. And as a maximum, okay, all of the previous is about maybe 9 marks. So you exceeded the needed mark. Okay, there are a free, I think in my opinion, a free or bonus mark that can be taken, okay, and can be considered in your bucket. For example, if you are talking about the repetition for the reliability of the experiment. So it's easily to mention in the last that a method by which you can repeat, okay, more than 2 times of repeat, it will be considered correct valid marking points, and you will grade it for this point by 1 mark. And the relevancy precautions, okay, you can mention any relevant safety precaution, and you're going to be graded for this by 1 mark. In some experiments, not all. I also, okay, say it again, in some experiments and not all, okay, you may use a control. Control is very good in order to, for example, be sure that the independent variable is very important, okay, the importance of it, better for comparison, okay, this is the use. And also there is another question based on this. So let's say it again. We have the variables, we have 3, independent, the one that we change, the dependent, the changing one according to the change in the independent variable, and then we have the controlled variable, or the variable that should be kept constant. Then we have the methodology, and we have a control, maybe mentioned last or maybe mentioned in the beginning after the variables, okay? And you have to mention the repeats. It's a must, okay? Because it's very, very important in order to avoid the anomalous data and also for the reliability of the experiment. And lastly, okay, you can mention any relevant safety precaution, okay? And what we mentioned now is more than about 9 or 10 marks. So it's easily to have or to gain the 6 marks in the designing an experiment, okay? Now I will unmute all of you, and if you have any question, you can ask. Yeah, it's okay. Any questions? Any questions? Any questions? You are unmuted all. Any questions? Okay, Mazen, go ahead. Mazen? Say it, Mazen. Okay, Mazen. Okay.
Hello?
Yes, go ahead.
Can you explain the control again? I didn't understand very well.
Yeah, the control variables, okay, are those that should be kept constant throughout the experiment, okay? That should be kept constant throughout the experiment. For example, if you are discussing the effect of temperature or on the activity of enzyme, you can say, for example, the same pH or determining the same amount of or same value of pH. For example, if you are studying the photosynthesis rate, okay, or the light intensity as an independent variable, and the dependent variable may be the amount of bubbles released. So you have to make, in this case, humidity the same. You have to make or have to put the temperature the same. You have to put the air or the wind speed the same. Any other variables that may affect the result. Okay, they are variables. Okay, they are in the environment of the experiment that should be kept constant. Okay, not to affect your results. Is it clear, Mazen?
Yes, thank you.
Okay, welcome. Any other question at this point? Any other question at this point?
No, I don't think so.
Okay, so we will go now for the second point, which is the table. Okay, I'll put you on the mute, then I will say some instructions and then back to you. Okay. If we back to the same exam that we did together, okay. Here, I think he asked you here to make a table, okay. Prepare a table for the student's results, okay, and measure the height or whatever the question is. Let's say about the criteria of the table, okay, the criteria of the table and how you will be graded based on the Cambridge marking scheme or the examiner reports. Okay. Okay. First of all, okay, the table, I have here also some responses. Okay. Okay. I'll check for another one. It's much better. Okay. Okay. For example, this is another question, the same with 3 marking points. The table, as I just mentioned, okay, may be from 3 to 5 marking points. Okay. And in one of the exams, I saw 6 for only one variant, which is very rare, but it may be happen. I can't let you see it now. But number one, the table have to be, must be ruled. Okay, must be ruled. Draw with ruled lines. Okay, with appropriate columns and rows. With appropriate columns and rows. This is 1 mark. Okay, second mark. Okay, this is the headings. The headings should be, or the columns and row should be with appropriate units in the heading, not in the data cells. In the heading, not in the data cells. Okay, this is also 1 mark. Lastly, the recording of the whatever he told you here, the color, whatever he asking you in the exam. This is the 3rd point. In some cases, it may be more than this. Okay, I'll let you see here, for example, okay, one of the responses. Okay, just a second. Okay, it may appear here. Okay, so if you look here, for example, to this table, okay, that came by 5 marks. Okay, by the way, this is responsive from your side. Okay, this is a graph, and this is a table of 6 marks, which is very rare to occur actually. Okay, by the way, the students is graded 2, okay, out of 6, but if you have a look here, okay, you can find out that, just a second please. Yeah, this is the table. Okay, number 1, the table should be, okay, ruled lines. Okay, he took a mark because he make a table with ruled lines. Number 2, the headings or the columns and rows, okay, should be with appropriate valid points in the headings, not in the data cells. Okay, he took also a mark because he put the units in the columns, but in some cases the examiner may not grade him a mark here because the columns and rows are not enough, because it's mentioned that there should be at least 5 columns, but in this case he just only do 1, 2, 3, 4. Okay, so this is maybe penalized, okay, by deducting 1 mark. So this is this point. Okay, actually, 1 second, please. Okay, let's back to the figure. Okay. Yeah, this is the figure. This is the table. I'm sorry. So number 1 was the table have to be with ruled lines. This is a mark. The headings should be the columns and the rows should be with appropriate units. Okay. Okay. In this case, it should have 3 trials. Okay, but he did not even given any attempt to this point, so he penalized by deducting 1 mark. And it should be 12 measurements, but also he did not do this, so he penalized by 1 mark. And he gave him here 2 conditions in order to complete the 6 marks. By the measurement in the light should be in between so and so, and even in the dark should be in between so and so. But he did not follow the rules, so he deducted. So he penalized. But if we can see, for example, a good attempt for a table of 4 marks, for example, or 5. Okay. Okay, 1 minute. Yeah, this is a good table. And also I did mark a lot of your tables was really good. Okay. Okay, let's discuss this. For example, here, the table, okay, should be drawn with appropriate, of course, lines, which are ruled. It's a must. And the number of cells also should be appropriate, which is already done here. So 1 mark given. Correct number of columns and rows, this is called headings as a marking point, should be with appropriate units. Okay. And the data, or coding the data correctly in the cells. Okay. All of the units, as we mentioned, should be in the headings, not in the data cells, or you're gonna be penalized. If he asks you anything else, he will ask you, or he will grade you if there is convergence, if there is any averages, for example. Okay, if there is any calculations should be done. Okay, you have to put those in your consideration. Now I will unmute all of you in order to take questions. Yeah, any questions? Yeah, all of you now are unmuted. Any question?
Yes, sir.
Okay.
I have a question.
Okay.
But can you just shift to the table a bit? It's not very clear.
Okay. Go ahead.
Sir, so it's okay to put two rows on top of each other. It says time seconds and then plant X.
No problem at all.
Okay.
Okay.
That was my question. That's it.
No problem. Okay. Another question. Yeah, another question.
Sir Abdurrahman.
Muhammad say for the experiment, if we should not say for testing in biology is not graded points, is no credit points, no credit points. Take it in consideration, Abdurrahman. Abdurrahman Muhammad mentioned in the chatting that for the experiment, if we should not, you should not say for fair testing. I asked the chemistry teachers and I did not go through the chemistry because it's not my specialty. Okay, but in chemistry it's maybe still accepted, but in biology, okay, it's mentioned penalize for fair testing. But you may say for better comparison. So I will. Any questions? There is a one that is making the volume so high. If you can reduce it, please. Any questions, guys? Regarding the table, are there any questions? Okay, I'll put you now in mute, and I will go through the drawings. Okay. 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 Now I'm gonna talking about the drawings, and the drawings is minimum 3 marks, maximum 5 marks. Okay, minimum 3 marks, maximum, okay, maximum 5 marks. Actually, they are always under OSDP, or in some cases, and most cases, OSDL. O is for the outline, S is size, D is the detail. Detail may be only one, and in some cases, it may be more than one. Okay, it may be more than one. More than one means maybe 1, 2, or even more, and even the one criteria may be more than one single detail together in order to take the mark. Okay, so in this case, for example, the student, he got 2 out of 4, but he asked him here to make a large drawing of the cross section. Okay, do not draw any individual cells, do not label the drawing. He didn't ask for labeling. That's why in the marking criteria there is no L or there is no labeling. Okay, so number one, the student here, he did a mistake, okay, by making the cuticle layer very thick. Okay, actually he shouldn't do it. He do all of this layer as the cuticle. Actually it should not, and so this line should be removed. Okay, those 2 lines have to be removed, and this one that I crossed right, it means that this is the epidermis layer. This is correct proportion, okay, and correct even as a layer. Also, this line should be removed because he thought that this between those 2 lines, this layer as the epidermis, and all of this thick layer as the cuticle. Actually, it should not. It should not be like this. So those 1, 2, 3 lines, okay, they should not be there. So let's go through this marking this specimen. Here for the outline, normally the outline should be clear, single, continuous, or unbroken, unshaded, not ruled, okay, not stippling, not hatching, okay, lines. This is the criteria, all of the criteria that may be mentioned. And in some cases, I saw some of you when they are drawing, okay, I know there are few, but still, they may use a compass, they may use a protractor, they may use the ruler. Actually, this is totally, okay, unaccepted and should be avoided. It should be avoided in your drawing, because you are drawing a living thing, and there is no living things with ruled, okay, or with a protractor or with a compass. So the criteria of the line that should be clear, single, clear, continuous or unbroken, both are the same, unshaded, never shade, never ever shade, you're gonna lose mark, and do not use all of your the ruler, the compass, the projector, and do not stipple or hatch the line. This is the criteria number 1. He took 1 for this. Regarding the size, outline is a mark in your bucket. Actually, you can just imitate the drawing. This is the clue. Imitate the drawing, and you're gonna take the mark. So outline is a mark in your bucket. Just imitate the drawing. Regarding the size, okay, it should be more than half of the space provided. Should be more than half of the space provided. And in some cases, he mentioned particularly, okay, a specific length for the drawing. Okay, such like the orange one that I mentioned, 80 millimeter, or which is more than 8 centimeter. In some cases, he did mention. But to be in the safe side, make it large. Make it large. And you're gonna take a mark. Make it large. Okay. Number 3, the details. Actually, details may be differ from one question to another. Okay. Here he asked for 4 to 5 layers shown. If we remove those wrong one that the student do, we're gonna count clearly 4. Here there is the upper epidermis, and underneath it there is the palisade, okay, and here we have the spongy layer, okay, or spongy mesophil, and here we have the lower epidermis. So they are 4, so will be graded for a mark. Okay, this is the only detail was there. The proportion here is mentioned as a graded mark, okay, or a credit, that the student should draw correct proportions, correct proportion. Of course, I did, okay, deduct a mark, okay. Here regarding the detail, the student, he drew 7 layers, okay, 7 layers, which is unacceptable, okay. If we count the layers, okay, 1, 2, 3, 4, 5, okay, 1, 2, 3, 4, 5, yeah, 6 or 7 layers. So it's not acceptable, okay. Regarding the proportion, as I just mentioned, the correct proportions have to be taken in consideration. If we up to see the picture itself, here we have about equal. It's not the same, but about equal proportions between the palisade mesophil and the spongy mesophil. A little bit more for the spongy. If you draw them mostly the same, it will be accepted. Okay, this is the upper epidermis. Okay, and this is the cuticle, but it's not a very thick layer. It's not a layer, just a lining. Okay, or just a cover. So the last one, okay, the palisade layer between third to half. This is the criteria as per the mark scheme. Okay, the palisade layer between third to half of the total mesophil. If he draw it half, it will be accepted. If he draw it 1/3, it will be also accepted. But here, it's not accepted mark. So I will put you now in unmute, and I'll take your questions. You are all now unmute. Any questions?
Sir, when it's like cells like this, do we always have to, like, you know, draw like draw its thickness or whatever?
The proportion. The proportion.
Yeah.
What is the proportion of the layer? You got it? This is the question here. What is the proportion for the layer? That's it. You got it?
All right. Okay. Yeah.
Yeah. Any other questions?
Sir.
Yes.
So for this question, all you need is 4 continuous lines proportioned correctly to get the mark?
Yes. Yes, as a minimum. They may be 5, but why they may be 5? Because if you look here, the palisade is more than 1 layer. Okay.
Yes.
There's 1, 2, 3. So it may be accepted, but 4 as a minimum. I have here a question from Abdurrahman Hatim. He say, when we write the headings but forget to put the wrong the wrong units, do we lose the mark? You may lose the mark. Yeah, because this is a very important point. Okay, because it's mentioned if he did not put the units, penalize a mark. Okay, this is a very important point actually. Okay, so you have to put this in your mind. Also, there is Muhammad's iPhone. Say for the cuticle, can we draw, okay, a thicker line? Okay, yeah, you can draw very small line, very thin layer. Okay, but it's not like drawn here in this attempt. It's wrong. Okay, how much time should drawings take in general? Not more than 5 minutes. Not more than 5 minutes. Okay. Okay. Go ahead. Okay. Okay. Excuse me. I have to take a call for just 2 minutes and I'll come back. Okay. Bye bye for now. Okay. I'm sorry for this. It was urgent call. Okay. Now all of you are unmuted. If you have any questions in the drawings, please ask. Now I'll talk in generally, and after this, I'll make another session for your drawings or your own questions. Yes. Any questions? Yes, guys. Any question? All of you are okay? Any question? Okay. If you do not have a mic, you can write in the chatting, then I can read it. Okay. Mazen asking, how can you show us how to draw this correctly? Okay. Let's do it. Let's go ahead. Just a second. I'll connect my tablet, my graphing tape, then I can draw. Okay. Okay. Now it's on. Okay. For example, this was the question. Okay. Let's see that I will annotate this one by drawing. So if you look here, okay, I will just minimize the picture in order to see the both. Okay. This is the drawing. Okay. or I can even make another one, which will be better. Okay. I will not draw in his drawing. It's better to do another one for my own. But just a while. Okay. Now all of you see the, you can see now the whiteboard I have. Can you see the whiteboard now? Okay. If all of you see the whiteboard, please give me an indication. I just want to be sure that all of you see the whiteboard.
Yeah, we can see it.
Okay, great. Okay, that's great. Now, if you have a look here. Okay. I can draw first the first line or the upper epidermis. It's like this. And sorry for my drawing, I'm just using a graphing tape, it's not that precise. Then we have to make a correct proportion for the epidermis, that it should be like this. You have to make a correct proportion. Okay, it should not be small here and wide here and small here. No, it should be the same. And here, okay, just 1/3 or about 1/3. You can draw a layer. This is the palisade mesophyll, and another layer here. Okay, this is the spongy mesophyll, and here there is a lower epidermis. So how many layers I have? First of all, how many minutes I took to draw? About maybe 3 minutes or 2 minutes. This is one layer. This is second layer, third layer, fourth layer. Okay. Now, is it okay, Mazen? Okay. Is it okay? Yes, okay, Mazen. Now I will go for another point, which is the tables. No, we did the tables, the graphs. Okay. Yes, let's take this for example. Okay. First of all, the criteria. Okay. Here he wrote a line that best fit, which is very, very common to come. And the graphs, the graphs or the bars may be from, they are varying from 3 as a minimum, 4, 5 as a maximum. Okay. And if you look here, you can see. Now, are you seeing both or just, I will close this. Sharing. Okay. Now I'm just sharing the PDF. Okay. 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 Now, if you look here, the criteria of the table. Okay. Number one, axis. Okay. Number one, the axis. Axis should be labeled. Okay. First of all, labeled with proper units. Labeled with proper units. Axis or the orientation of the axis should be put in consideration, and it's very, very, very important. Okay, the independent variable will be in x-axis and the dependent variable on y-axis. The independent variable on the x-axis, the dependent variable on the y-axis. Okay, this is the main point regarding the axis. Okay. And the units. If you do not put the units, you're going to be penalized. You will be penalized. Okay. So here we have point number one. Number two, we have the scaling. Okay. It should be even. The data have to occupy more than half of the grid. Okay. This is 1 mark. Scale should be even, and the data should occupy more than half of the space provided. Okay, should be. This is not 1 mark. The plotting the line, all points plotted accurately. The error that may be considered is not more than plus or minus half small square. Plus or minus half small square. This is 1 mark. Regarding the line, which is maybe a little bit hard for many of the students, the line, okay, should be best fit. Best fit. Yeah, what's the meaning of best fit? It should not pass by all of them. No, should not pass by all of them. Okay. Is it? Is it should be away from many? No, it should be smooth curved freehand. Should be smooth curved. Okay, freehand. Okay, or uh, just a second. Okay. Okay. Now please listen up. Just a second, please. Okay. Just there's a sound. Okay. So the line should be smooth, curved, okay, freehand, or point to point. I think point to point in those that may not have that skill of drawing, okay, maybe good. Okay, point to point by ruler will be good. Okay, and also will be graded for a mark. Okay, so this is the criteria of the line that best fit, which is frequently come. Also, we have, for example, from your uh, drawings, okay, the last thing in the graphs, we may have a bar chart. Okay, I'm just looking for one of the bar charts. Yeah, this one, for example. Let's talk about it. Okay, what are the criteria of the bar chart? Okay, number one, the axis, it's same. Okay, labeled with valid units. Okay, the independent should be in the x, the dependent should be in the y. Okay, and maybe the dependent will be in the y, and x you just write the name of the changing things, or the one that you are changing by your own, that are considered also as independent variables. Okay, the scale also have to be even scale. Okay, okay, even means with ruler. With ruler you have to do this. Okay, and to be appropriate, and plotting appropriate, all of the points have to be plotted in correctly. Okay, and to fill at least half of the space provided, or half of this grid. The plots have all the points that you will draw, all of them have to be plotted accurately. Okay, to the error of plus or minus half small square. Okay, regarding the bars, they have to take in consideration some points. Number one, with equal width, so the space between the bars have to be the same, exact the same, not touching. Never do them touching, because if they do so, it's a histogram, it's not a bar chart anymore. Okay, and also, okay, with equal space between them, or a gap in between the bars have to be the same. So the same width, same gap, okay, and not touching. Those are all the criteria of the bar chart. Okay. Now I'll open the discussion and take your questions. You are all now unmuted.
Sir, can I ask something?
Yeah.
So like, not in bar charts, but like, like the ones where you have to draw lines, right? If the y-axis values start from like 500 and end at 1000, is it fine if we draw like a zigzag line at the
Yeah,
starting of the
that's
y-axis?
right. Yeah, you are right. It will be like this. Okay.
All right.
It will be like this. For example, I will be sure that you are sharing this. You are sharing this. Okay. You are sharing the whiteboard. Look, if you are drawing it as you just mentioned, and the values are so high, then you can just poke this line, for example, like this. And then you can start, for example, 2000, okay, 10, okay, and 2020, and so on. Yeah, another question? Yes, another question? Any other question?
Sir, for the same question Hassan asked, it says line of best fit, but it went through every line, and he got 4 marks.
Point to point will be proper.
It went through
I
every
know,
point.
I know, I know. Yeah, I know.
So isn't that wrong? Because it said line of best fit.
Have to be straight. You may do it. You made it straight, but if so, it will not pass, or it should not, or it may not be passing by all of the points, which is not a problem because you may have 1 point above, 1 point below. Okay, you may use point to point as the instructed. Okay, which is easier for persons that have hardness in the drawings. Okay, this is much safer. You can also do the smooth curved free-handed, okay, line, which is also accepted. All of those are accepted. Any one of them. Okay. Any other questions? Any other questions? Okay. Any other questions? Okay. For today, we finished the number 1, how to design an experiment. Number 2, we did together the drawings. Okay, we go through the graphs, and also we did the tables, how to do them. So all of those consider more than half of the marks of paper number 6. And as I need to see some of your responses that you did show me, and I did mark